In the previous video, we have discussed that the wild type cells or the single knockout of Calnexin or calreticulum, the GPI-APs are able to retain majorly within the endoplasmic reticulum, and very small fraction of those GPI-APs are able to express on the cell surface. However, in the case of dual knockout, we have seen that there is a loss of quality control of endoplasmic reticulum, and GPI-APs are able to express on the cell surface. In this video, we will discuss what kind of GPI-APs are able to express on the cell surface of this dual knockout. So from the heading, it is clear that the final results that they have observed is that acetylated and misfolded GPI-APs are able to express on the cell surface of Calnexin and calreticulum knockout. This is the dual knockout. So we will discuss how they have come to this conclusion from the experimental results that are presented here. So in the results that are presented on the left hand side, they have used the wild type cells. So in this, in order to introduce this artificial ER stress, they have used thepsigargine. So when they use the thepsigargine, along with this, they transfect the cells with this CD59 with the flag, with the EGF flag reporter, and the CD59 is the mutated CD59. That is, it is mutated at C94 with S. Simultaneously, they have also transfect this MRFP KDEL sequence to check the position of this endoplasmic reticulum. So after 1 hour treatment of thepsigargine, they can see that the most of this misfolded CD59 is retained in the endoplasmic reticulum, and this is being confirmed quantitatively using this Pearson correlation coefficients. And after 3-hour treatment, they can see the spread of this misfolded CD59 on the cell membrane, and same things are being quantified using this Pearson correlation coefficient, and they can see that the statistical significant difference is there between 3-hour treatment and 1-hour treatment. After 6-hour treatment, there is not much difference. However, it maintains the statistical difference with the wild type results, with the 1-hour results of the thepsigargine treatment. So in order to validate that this kind of phenomena exist, in order to check whether this kind of phenomena exists for GPI-APs or for the other ER resident proteins also. So they stain the some ER resident proteins such as Calnexin, HLA1, 147, and check them after 6-hour treatment. After 6-hour treatment, they are not able to see that this intracellular endoplasmic reticulum resident proteins able to migrate to the plasma membrane, as the signals of this KDEL is overlapping with this flag signals or EGFP signals of this Calnexin and this HLA1. Calnexin and HLA1 that can be seen here. KDELs is in line with this panel C is in the panel C. They see the signals of this Calnexin and this EGFP, and they can see that they are not overlapping to each other, and this CD59 is able to migrate to the plasma membrane. And for this panel D, they checked it for this intracellular proteins such as intracellular protein of endoplasmic reticulum. So that is HLA1, 147 flag and KDELs, and they can see that overlap of these KDELs and this intracellular endoplasmic reticulum protein. And they also check it for the CD59 that has this transmembrane domain. So after 6-hour treatment, they can see that this CD59 with this transmembrane domain is able to retain within the or migrate. We can see that some kind of migrations is able to migrate at the cell surface levels. So quantitative analysis of this panel E is not shown, but it is only mentioned that the transmembrane domains are able to retain this the CD59 with the transmembrane domain are able to retain within the endoplasmic reticulum, and that is what is being understood from this figure, that they are not deviating too much from this KDL sequence. And after validating that this kind of phenomena, that is at small ER stress, the GPI-APs are able to express on the cell surface even on the wild type. They need to check what kind of these GPI-APs that are expressed on the cell surface, whether they are different from GPI-APs expressed from the dual knockout or they are similar. So when in the wild type cells, what they have used, they use this mutated CD59 and this CD55, and they can see that that even after the 6-hour treatment, there is a decrease in the PIPLC. There is a decrease in the or the remaining amount of the CD59 after PIPLC treatment is reduced and are similar to each other, even for the CD59 and also for the CD55. However, for the dual knockout, they do not see decrease in the signals with the PIPLC treatment. So here in the dual knockout, the GPI-APs that are expressed on the cell surface or on the plasma membrane are not PIPLC sensitive. However, in the case of wild type, they are PIPLC sensitive, and these things are quantified in this panel B, where they can see that in the wild type treatment of cells, the misfolded expression of this CD59 is minimum in the wild type without any thepsigargine treatment, that is shown by this dark border, and after thepsigargine treatment it is shown by this dark border in the bottom panel. And here we can see that these results are statistically significant in this quantitative analysis. However, for the calciticulum knockout, there is not much increase, and they are almost similar. That is, increase in this misfolded GPI-APs expression on the dual knockout is non-significant and is not affected by the treatment of thepsigargine. However, for this CD50 and similar results they have observed, not that much similar but slightly changed result they have observed for the CD55, where the difference for wild type and where the difference for wild type with thepsigargine treatment and without thepsigargine treatment is statistically significant. And however, for the calciticulum and Calnexin dual knockout, the difference for expression of the GPI-APs are not statistically significant related to each other, but related to the wild type they are statistically significant. So results are similar in both the cases. Yeah, that's what I have said. And one of the hypotheses that they think that why they are not so much sensitive to the PIPLC in the case of this dual knockout, however they are sensitive to the wild type, is because of the retention time of this misfolded protein in the wild type reticulum, in the endoplasmic reticulum, in the wild type cells in the endoplasmic reticulum. Let me repeat this statement. One of the reasons that they thought is that retention time of this misfolded CD59 in the case of wild type in endoplasmic reticulum is high compared to the dual knockout. So the wild type cells, they get chance for deacylation, whereas in the case of dual knockout they do not get chance for deacylations. And since the cells misfolded proteins in dual knockout is not deacylated, they are not sensitive to the PIPLC, whereas in the wild type cells they are deacylated, so they are sensitive to the PIPLC. So the sensitivity of this PIPLC is shown in this panel C and panel D, For this wild type cells, we can see that in the wild type cells is considered as a reference one, whereas in the case of dual knockout, they can see there is a more remaining of this misfolded GPI-APs, and this difference is statistically significant for the CD49 for the CD59 mutations, and similar results that they are observed for the CD55 mutations. And in the panel D, what they have shown is that they have shown these results as remaining of misfolded GPI-APs after PIPLC treatment. And in this case also, I think the panel C and panel D looks similar. That is, they represent the same results. That is, in the case of this dual knockout, there is a more remaining of this misfolded protein, indicating that the acetylated misfolded proteins are expressed on this dual knockout that are not sensitive to the PIPLC. And same things they are observed for this CD55 misfolded CD55 cells. So from these results they made a point that it's the acetylated and misfolded proteins that are expressed on the cell surface of this dual knockout. Let me again check the panel C and panel D. What is the difference between panel C and panel D? So I think one of them could be thapsigargin treated and one of them is without thapsigargin treated. However, we can see that the results are similar. In this figure, it is not shown which of them is thapsigargin treated. However, if you want to know about this in detail, you can go through the reference where you can check the caption of these figures. And if you need to understand more about this figure in detail, you can refer this reference link from where this experimental results has been taken. So thank you for your time and have a good day.